Is it now on?
Okay. Oh, now you're on. Very good.
Okay.
Thank you. I'm going to turn it over to Dr. Joan Rose. I apologize for the sound audio difficulty there. And our next presentation is Dr. Joan Rose from Michigan State University on microbial responses to land physical, chemical, environmental, and hydrological factors. Thank you very much for being with us today.
Oh, thank you very much. And yeah, it would have been hard to do the presentation without the sound there. Okay, well, I want to acknowledge my co-authors on this. Mark Verhoostrate, who just finished his PhD and is now at the University of North Carolina, Dr. Dave Heineman, who's the hydrogeologist, and his team, Sherry Martin, and Dr. Anthony Kendall, who did a lot of the other work here. I'm a public health water microbiologist, and I deal with water quality and how it affects human health. And we got involved in this project really to think about whether we could develop tipping points for human health and waterborne disease endpoints, which really link the idea of water quality and our own health and the health of our ecosystems, our beaches. And will this improve our understanding of the causes of the problems with water quality and assist with science-based decision making? I mean, basically, I get asked this question all the time: how safe is your water? And that's always it seems like a simple question, but it's really difficult to answer, and it's getting more complex in this era of the Anthropocene that we are living in. Now, we know we have concerns about our coastal systems, and in fact, some recent analysis suggests that not just in the Great Lakes, but all over the world, over the last 100 years, we're seeing a degradation of our coastal waters. Higher eutrophication, more nutrients, more beach closures. What you have in front of you is a map of all the recreational outbreaks in ambient waters. This doesn't include swimming pools and spas. There's been about 157 outbreaks in the last 20 years, and they're almost all associated with lakes. There's a few associated with rivers and a few associated with marine beaches and some springs. And you can see that the East Coast in the Midwest has their share, and we have a lot of lakes. So, and there's a lot of different types of waterborne pathogens. Includes viruses, bacteria, and parasites. We're seeing issues that we normally wouldn't even put on this map. Things like swimmer's itch show up at much higher prevalence. So these are, an issue. And so obviously we don't want people to get sick. We've had a number of outbreaks where people actually have gotten sick in the Great Lakes, but it's not just that, it's this increasing beach closures. I think the latest outbreak, or large outbreak, that we had in Michigan was this outbreak at the Kent County Millennium Park. And you can see that, in our summers, a lot of people like to use the waterway. It's a big economic driver of these communities. And this happened to be a norovirus. And even when they closed it down and thought they had taken care of it, when they reopened it, people started getting sick again. So the contamination stays around. And we're changing the nature of our interaction with our waterways. You can see that we now have these parks that have all these water features, which are quite fun, but may be difficult to clean up once they're contaminated. We also know from some studies just recently published that it's not just about swimming. There's evidence that boating on Chicago riverways and general waters was associated with gastrointestinal illness. So these are effluent-dominated waters, and they were associated also with skin infections and, in particular, eye infections. And there was about 13 to 15 infections per 1,000 boaters. We found human enteric viruses. They ranged at pretty high levels and pretty high copies. So we know when wastewater and sewage and stormwaters and CSLs, which are carrying these pathogens, enter into the waters, that people will get have a possibility of getting sick when they use the waters, both for swimming and for just fishing and boating. So we have a number of threats we've identified. Obviously, those sources that bring fecal contamination, both from animals and humans, are high on the list. Sewage, septic tanks, combined sewer overflow, stormwater. We have a lot of inadequate infrastructure that we're going to have to decide about fixing. And we also have internal risks that are starting to show up, toxic algal blooms, and these are exacerbated by climate change. Obviously, the consequences are disease, and not just these outbreaks, because we recognize that outbreaks are just the tip of the iceberg, that most of these are underreported unless we do these specific epidemiological studies. And I think the other thing that we're realizing is it's not just acute effects. That is, you're sick for a couple of days and you get over it. We're seeing more and more reactive arthritis and chronic outcomes associated with salmonella and campylobacter infections. And this is being well documented now, that waterborne exposure to these pathogens can lead to these chronic health problems down the road. We know about these long shorelines, and the Great Lakes has a huge coastline that's important. And these coastlines are not only important for our health, but the economic health. And estimates are that shoreline counties contribute $5.7 trillion to the U.S. national gross domestic product and 48.6 million jobs. And you can see that while we do pretty good in the United States in terms of access to sanitation and wastewater treatment, the rest of the world is really struggling. So we want to protect what we have, restore where we have problems, and look to the future. The beach closures themselves, it's estimated in the Great Lakes region that a single day, anywhere from $30,000 to $230,000 can be lost by the community. And so these are really real significant concerns caused by water pollution. Now, we monitor our coastline, and we have a system in which we've monitored recreational waters, which began early in the history. We use methods in which we bring the water sample into the laboratory. We filter it, usually volumes of about 100 liters, or we at least assay 100 liter, or 100 mil volumes, I should say, not 100 liters, 100 mils, sort of a glass of water size. And we look for what we call fecal indicator organisms. These are bacteria that live in the intestinal tract of animals and humans. We have established that a relationship between these fecal indicators and water quality and disease outcomes. Now, total coliforms on the bottom, that's used mostly for drinking water. We don't use that for recreational waters. And where we've moved for recreational waters is E. coli. And we have both most probable number methods, as you can see with this tray, where you dilute out the sample and you count the number of squares that show up positive. And we have what we call colony counting methods, where we filter the water, put it on agar, and we can actually count the number of bacteria that are alive. And this next slide actually shows over time the various studies that have been done, and these have been done in marine waters, fresh waters around the world. The relationship between fecal indicator bacteria, which is on the concentration on the x-axis, and the potential or odds ratio, that is the likelihood that you're going to get sick, on the y-axis. You can see there's a lot of different curves, and this is no surprise. These different curves are associated with the geomorphology of the beach. You know, how the beach is situated, what kind of volumes, how does the rain and rivers influence the beach, and the land use around that beach. So it's no surprise that we have a lot of different curves, but ultimately in a meta-analysis we've been able to come up with a level of bacteria, fecal indicator bacteria, below which we believe that people are relatively safe when they swim. and that there's very low risk of getting disease or getting sick when you swim. So we have what we might call a threshold. That is and it's a regulatory threshold. It is based on this science, which builds the relationship between the pollution and the risk of getting ill. But one of the problems we have is that while we have developed this threshold, we've developed an approach for monitoring and where we want to be safe, where we want to be in terms of safe, the science has moved on. And basically, we want to know when we have these pollution events, or when we have chronic beach closures, what is the source of the pollution so that we can fix it? So that's one question that's been asked since we started using these indicators. And we never could really answer that question before. What is the source of these E. coli and these pathogens that are making us sick? I mean, in some cases, like the Chicago River, where it's receiving these effluents from the wastewater, it's pretty obvious it's effluent dominated. But at most beaches along the Great Lakes, we really have a mixed land use, and we don't know the source or the predominant source of the E. coli and pathogens. But we're starting to develop these tools that will allow us to look. The other thing we've recognized is that these sources carry different types of pathogens. There's been emerging pathogens, things like parasites like Cryptosporidium and Giardia, new viruses like the polyoma viruses that are of great concern. And we know that our fecal indicators don't always give us the right answer there. So we're looking for new tools and using new tools that will give us an idea of what's going on. So it's these new tools and techniques that will help us spend our precious dollars. We want to be able to make better decisions. And we want to understand, are there tipping points or these areas where the sources of these contaminants are coming from, where we can do something about them. And we think about land and our land use as the source. And here you see two maps. On the right-hand side is a map of some of the sources. This is the sewage treatment plant discharges in Michigan. Every place there's a little dot is a sewage treatment plant discharge, and this is a varying effluent quality, carrying varying levels of parasites and viruses and bacteria. These all flow to the Great Lakes eventually, or one of the lakes. And on the right-hand side, you see the impact. That is all the beaches, the 606 public Great Lakes beaches that we monitor in Michigan. And so here's an example of one of the sources and some of the impacts. And so can we use these new tools to make better decisions, to understand, are there areas of demarcation, that is where there's a significant change in the water quality based on these sources. And so can we use these microbial source tracking tools? Can we use risk assessment models and predictive models to get a better handle on this? So our objectives in joining this team, that we're, this team of scientists, that we're looking broadly at sort of the idea of land use, climate, impacts, threats, using these new mathematical approaches and trying to understand, are there demarcations, are there areas where there's significant rate of change. We joined them, and we identified two objectives. One, to examine the occurrence of E. coli, our conventional indicator that we use, our fecal indicator bacterium that we use in recreational waters and ambient waters, and compare it against one of our new source tracking markers. And in this case, it was a human sewage marker, the Bacteroides theta, in river systems under distinct hydrologic conditions. We wanted to identify specific land use patterns contributing to this idea where there was a dramatic change in the rate of pollution with human waste sources. So we use the conventional IDEX coli alert tray. This is a most probable number. You divide up the sample into smaller volumes, and depending on which cells come up positive and which cells come up negative, you can actually quantify the concentrations of E. coli per 100 mils. That's not source specific, as I mentioned before, but it is the indicator of choice in Michigan. We do use it to say whether a beach is safe for swimming and whether it could be open or closed or placed with an advisory. We also use this Bacteroides theta. This is we've done some studies on this, both in sewage and in animal waste. We have 97% human specificity, so it really gives us very good linkage to human sewage. It's a DNA-based method. That is, we take the water sample, we filter it, we capture all the cells, we extract the DNA from those cells, and then we use polymerase chain reaction to amplify or copy the specific gene target that is associated with this Bacteroides species, B theta we call it for short, and then we can actually quantify it so we can get concentrations. Now, the qPCR method does not give us viability. It'll detect both live and dead cells, but one of the things about this B theta is that it doesn't regrow in the environment. Once it comes out of the human body, it's no longer capable of replicating, and so it's a steady input as an indicator of fecal pollution. And unlike E. coli and Enterococci, where we have the ability of those bacteria to regrow. It's part of a very large family of bacteria that's found in animal and human guts at very high concentrations. It's anaerobic, and that's why it doesn't survive long. As it's exposed to air, it starts to die. So it doesn't reproduce outside the host. And it is difficult to cultivate, but we can measure it by this qPCR method. And it's a very good human sewage marker. We've just published, we've just participated, and the group has published a blind round-robin study in which a number of different types of samples, both human and animal samples, were put together and sent to the labs blind. We analyzed those samples, we sent the results back, and we were quite pleased with the way B theta. And so we're starting to see some validation, a national validation of these markers that we think we could utilize, at a state and national level with some confidence. We also used, of course, we're looking at land use, so we use the ESRI ArcMap. We use Sigma plot. We used R package that was already presented with and used the classification regression tree analysis, this card analysis. So we're all trying to use similar statistical approaches to look at whether we can find these tipping points. So let's take a look at the study. It was a statewide study. And 64 rivers were sampled. Not only was E. coli and B theta examined in these samples, but nutrients were collected, ions, stable isotopes, chlorophyll A, epiphytic algae, of course the pH, conductivity, temperature, dissolved oxygen, precipitation was measured, point source nutrient loads, and river discharge. Now, I think the interesting thing about this design is, with water, we're always thinking about time and space. What we did here is spatially, we collected samples as a spatial composite at the bottom of these watersheds. And so we spatially collected the water that was coming from these watersheds at the outflow. And then we took temporal samples. So we took a number of temporal samples of these 64 river watershed systems. So I think it was a very interesting design. Here's what the land use looks like in these watersheds, and we had very good coverage, as you can see, of the lower peninsula. So sampling the water that comes from really a very large percentage of the land mass in the lower peninsula, and covers both urban and rural and forested and agricultural land use categories. We also started not just looking at land use coverage, but land use characteristics. And I think this is very important because land use characteristics give us some resolution of these sources. And we're going to have to do more of that in the future. So this gives you a map of some of the wastewater treatment plant infrastructure. It's sort of another look at that map where I showed you the discharge sites. And in addition, we looked at, of course, the population distribution, mapping that out because we're looking at human sewage. And using these two, we were able to actually estimate a septic tank system density. If you know some of these land use characteristics, like how many heads of cattle are in these watersheds, or this, this type of information, if we want this source allocation information, it's very difficult to get, or you have to go straight to county boundaries and look up the data at each county level and try to put it together. Often the data is missing. Here we've come up with some approaches in which we can subtract out population, our sewer and wastewater infrastructure, and come up with the septic tank density. And this was ground truthed in a number of counties, and it looks to be quite good. So now we've got some idea of the sources of human sewage. We've got characteristics, land use. We took 3 temporal samples: base flow, spring thaw, and summer rains, which no surprise are quite different in the lower peninsula of Michigan. So let's take a look at some of the results quickly. So this is base flow. We had a lot of E. coli, only 4 non-detects. And the colors on the map give you the concentration. So of course the red being the highest concentration. These are in logs. So 2.9 is almost 3 logs, which is 1,000 E. coli per 100 mLs, down to non-detects, which is the very dark green. Our geometric mean across the lower peninsula is 1.4 log, and you can see, no surprise, some hot spots around these high population density regions along the coastline, and in the middle lower portion where we see sort of lower densities up at the top of the mitt, at the very top, I guess, of the lower peninsula there. Now, what did it look like at spring melt? It goes down. We have more non-detects, but not anything significantly. We do have more exceedances, however, of the EPA criterion. The map doesn't look too much different, except that we see that there's some areas that start to look a little bit more polluted. That is probably a result of this runoff towards these low-lying watersheds, and a dilution of what we're seeing in the middle portion of the land use. And finally, in the summer rains, we see it go up. We see the concentrations actually go up. So, of course, an indication not of perhaps this lower dilution of E. coli, but of this runoff off the land. So the concentrations go up to almost, up to almost 2 logs. And you can see that the levels of lower quality water are diminishing. We have no non-detects during this time, and we have about 10 exceedances. So I wouldn't say that we can say that our criteria for exceeding, this level of water quality safety changes during these 3 events, these 3 hydrological events, but certainly there's a shifting of where the concentrations are. Oops, let's see, I think I skipped a map. Okay, B theta. So let's look at B theta. Now base flow, I should mention that base flow, it's kind of interesting because Dave Heineman has looked at this. Under base flow, 80% of the flow or higher is coming from groundwater. Now B theta, we see a little bit different picture. The concentrations are much higher. Remember that this is a DNA marker. And it's a total concentration of the pollution coming in, and not of that which is alive. And remember, E. coli is killed during some of the sewage treatment system, whereas B theta would not be killed under sewage treatment. So under base flow, however, this is coming from flows from wastewater plants, flows from probably we're guessing from the groundwater. Our geometric mean is 5.1. We can see these hotspot watersheds. And the interesting thing is that overall the data correlated with septic tank system counts. And so we had a pretty high R squared, which was quite surprising to us. We weren't expecting to see this. But it did not correlate at this stage with the wastewater flows. Now, spring, melt, and summer, we got a dilution. We got a dilution of the B theta. And remember that both spring melt and summer rain, the E. coli was you could see the E. coli coming up. That's a cultivation rate, cultivational signal coming off the land. We only see a couple of hot spots really for the B theta in spring melt and summer rain. So we're just seeing this being diluted out below our signal as it comes off the land. This gives you sort of a comparison of our E. coli and our B theta across the seasons. So you can see that the E. coli was responding to flow, going up as we got more flow, base flow spring and summer, whereas the B theta was coming down. And so we do see these seasonal differences. They are statistically significant across the land, and this suggests this potential for overland flow versus this flow from potentially polluted groundwaters coming from the septic tanks that we see with the B theta. And the ability of rainfall to actually, in stormwater, to dilute out some of the wastewater signal. This gives the loads, similar picture. The previous was concentrations, and this is a loading over the time. So, you know, a similar picture, maybe not as dramatic in terms of the loading for the B theta, which probably is reflective of the wastewater contribution. Loading staying fairly constant, where some of the flows are coming in. But we still see an increase, a significant increase in E. coli loading to the system. So what about these threshold or tipping points? Can we get at something that could be used for better decision making? Because in the long run, we're going to have to make decisions about what we do on the land, about these sources of contaminants that are impacting our water quality, impacting our beaches, causing eutrophication, causing beach closures. So can we use these new statistical approaches to take a look at this? So we use the CART, and this is the B theta and the septic tank system regression that I showed you before, with a fairly nice correlation. And we were actually able to use CART to see where there was a change in the slope. That is, mathematically, was there a tipping point with a mathematical change in a relationship between increasing risk of pollution associated with the concentrations. And you can see on the left-hand side that blue line is pretty flat. When you look at the septic tank systems, which is on the x-axis, and the B theta on the y-axis, whereas we've got a change in the mathematical slope, which we are delineating as this tipping point. And so now we have a number of septic tank systems in a watershed that we could say, if this watershed is starting to reach this level, this may be viewed as a tipping point in terms of increasing the rate of human sewage impacts to your water systems, and that this may allow for people to make better decisions on when to change strategies for the density of housing, the density of septic tanks, or the use of wastewater treatment and advanced wastewater treatment. And here's the numbers. So more than 1,621 septic tanks in these watersheds was a concern. So the base flow, here we have this decision point now. We have these upstream lakes contributing also to B theta, and we believe that this is from high cottage densities around the lakes. So we were able to see an impact of lakes, and we need to go in and further evaluate that. And as many of you can appreciate, in these recreational areas, as they've grown and more people have put in houses, second houses, or moved out to these areas around some of our lakes, that we do have more higher density of septic tanks. Oh, my phone is ringing. Oops. Oh, I had one more thing. Okay, I just hung up my phone, whoever was calling me. I will call you back. You know, the other interesting thing was this was number of septic tanks, not density, as a key variable. And density has always been used, and so we need to sort this out a little bit more in terms of the different sizes of the watersheds, which watersheds had lakes and which didn't, and you know, to move these things forward. Okay, so that's where we are right now. We're doing further studies to try to understand this human signal, this human sewage signal. We need to do a little bit more from wastewater plants. We do know what the loading is from some of our sewage treatment plants. We do believe that these bacteria are killed after disinfection, if the disinfection is adequate. So we need to do a little bit more there, and we need to do a little bit more around these lakes. Lakes are also act as reservoirs of deposition. We do get the deposition of these bacteria, and lakes have an ability to take things out, as opposed to rivers, which stir things up. So that's the future, and we also hope to use some of the bovine source tracking markers we have to look at some of these watersheds. So thank you very much.
Thank you very much, Dr. Rose. And we are right at 2 o'clock. If anyone does have a quick question, you can go ahead and type it in. And I wanted to also make sure that people have on the schedule the third session, which will focus on nutrients. This will be on Tuesday, October 1st from 3 o'clock to 4 o'clock Eastern Time, using the same Adobe Connect site.
And people are free to email me as well if they have other questions later.
Excellent. Thank you very much. And I will send out the webinar direct link to the recorded presentations. We are editing these for future use on the Tipping Points and Indicators website, but for now, people can at least view the full presentation using the direct link. And I'll send that out directly after the presentation. So thank you, Dr. Rose, and thank you very much, everyone, for attending today. And we will see you all and talk to you all next week.